Okay, here we are. I have my list of class names. So I'm gonna start doing what I was hoping to start doing, which is A, continue practicing my names, and B, just you guys in recitation section 4 really get the shaft because I don't really get to know you. So this is the best I can do. What I learned is there's something like 97 people registered. And those 2 people have never showed up, so they don't count. 2 people are me and Dr. Texel, so we don't count. But still, there's a lot more people. Who has onions? I have an onion aversion. Actually, do you guys want to talk about onions for a minute?
Yes.
I
Love
actually
onions.
really like it.
No, but so here's the deal. This is what I realized recently. And tell me if this makes sense to you. I can't stand the smell of onions. Well, I didn't realize that recently. I've known that my whole life. But recently, like, I'm an adult, I power through things I don't like because sometimes the whole picture is like delicious. Like cilantro, hate cilantro, but I love Indian food and sometimes it's sprinkled on top and blah, 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 blah. But, okay, onions. I ate raw onions because I was trying to like feel the whole picture, and boy was I ill. I was so ill like for like a whole day. And so I was thinking that like maybe people who are upset, and then I read that some people have sensitivities to some compounds in onions that just make them feel ill. And so I was thinking maybe just over millions and trillions of years, people who are going to feel sick tend to hate this, like they have like this horrible aversion. So I want everyone out there who hates onions to go eat a raw onion. Let me know how it goes. Just email me. Say I was ill, I was fine, I wanted to vomit. Okay. Anyways, that's it for the onions. Okay. So that allowed other people to come in. That was fun. So here's what we're going to do. I'm going to start calling people randomly, but this is how it's going to go. I don't want you to feel scared, but I also want to avoid having the same 5 people answer all my questions because that can sometimes feel intimidating to other people. But so if I call on you and you don't know, you can ask a friend. So choose wisely who you sit next to. Okay? So you're not even alone. It's not even your point. I'm not saying on the exam you can ask a friend. But so you're not alone. The point is you're not alone. Okay. So I just want you to know that. So don't be scared that I'm going to call on you and you won't know. Just sit next to someone who you think will know. And the print is really small on here, so I might not be able to read it. Okay. So this is the last week of lectures before the exam. I've had some people say, is this included on the exam? Yes, this week's lecture. This is why I postponed it for a week, because I needed to cover this stuff. So this is our list. The only thing I've added since the other day was endocytic pathway, recycling endosomes, receptor-mediated endocytosis. So is that topics? Notice they're all in the same category of proteins getting to places. Quick business, exams on Wednesday. Who thought the exam was today? Who studied all weekend? Well, you should study all weekends anyways. Good job. Okay. But if you thought the exam was today, I'm sorry that you guys have been living under a rock. So the material's through Wednesday, and the exam, I keep getting what the format. It's multiple choice, and I'm going to do a short answer extra credit. So if you want to show me your brilliance, you will have that ability. Lecture 7 should be in the process of being up. The problem is with this very cheap app that I have for recording lectures is that sometimes something funny happens to the file, and I have a hard time exporting it. Like it just fails over and over again. So then I have to use a different program. And so 7 should be hopefully up by end of the day. It's happening right now. Okay. So this is where we ended the other day. Receptor-mediated endocytosis. We were talking about that. Remember? That seems like forever ago. Oh yeah, there was a Stump the Teacher extra credit. Most people got it. So check there's a column in Grade Center with cumulative extra credit points. Some people are starting to pile up extra credit points nicely. And it was just the question was how do viruses get into cells? But the real question was what's the mechanism for viruses using caveolae? And some people found some mechanisms of receptor-mediated endocytosis, basically, and then some clathrin-dependent mechanisms. That was very nice. And then some people just told me what I told them in class. And if you do that, you don't get the points. Okay, so we were talking about receptor-mediated endocytosis, and that just means that this specific endocytosis, and we have the examples of the LDL receptor and transferrin receptor, and I had a question over the weekend of, do we need to know, like, I don't know anything about insulin pathway, do we need to know all this detail? No, and these are examples. They're examples. So just understand the difference between the two. Compare and contrast. Understand the general differences, not specifics about LDL. Just understand the differences. Another reason for receptor-mediated endocytosis is to downregulate the receptor, right? And so this is what this is showing here. You have some receptor, and actually it's a GPCR. We'll learn about that shortly. And then you have, oh, that is a really annoying light. Yeah. Woo! Sorry. The person before us uses the chalkboard. So you'll have endocytosis, and there's your clathrin-coated pit, and then you go to your endosome, and then sometimes you can have recycling, but sometimes you can go to the lysosome. And so the reason you would want to send the receptors to the lysosome would be because you're trying to take it off of the cell surface because you want that signaling to stop. Remember, there's all sorts of receptors that do all sorts of things. Sometimes they're bringing in things from outside the cell. Sometimes they're binding to things and signaling. Sometimes they're communicating with other cells. And when you want to shut that off, really the way to do it is to send the receptors to be degraded. And so that's one thing that can happen here. So the question I have to remind you when you're studying, why would the cell want to send the receptor to the lysosome? I just told you, it's usually to shut off the process. Okay? Shut off that signaling pathway. Okay, so now we're talking about down-regulating receptors. And I do want you to kind of keep this all in mind as we go into signal transduction. So they can also be, receptors can also be down-regulated through sequestration. What does sequester mean? I'm gonna start my new thing here. I can hold this to remember. Taisha Marshall. Where are you? Oh, and the other fun thing is that when people aren't here, I don't know. Not that I take on this. Is Taisha here? Okay. Janine. Janine? Oh, he's here. And the best thing is they're going to listen to this later and feel embarrassed. Steven. Steven? Sits back there usually. Oh, my God, no one loves me. Hey, Ginger, I saw you before. Yay! I'm going to start having candy that I throw to the people who are here. Okay. So what does sequestration mean? Just basic definition. And you can ask a friend.
I'll try to make something out of it. I think it goes in sequence.
Let's try again. Okay. Let's ask that friend. Sequester. What does sequestration mean when we're talking about the economy?
Okay.
Self-contained drugs.
what, Kim?
The cells come together.
Okay, something's coming together. Let's keep trying. Does anyone want to add to that or change that? We've talked a lot about sequestration in the economy. Yes.
Setting something apart so that it's not with other parts.
Okay, yeah. So it's setting something apart, so it's hiding it from other things or keeping it away from other things. When they talk about sequestration in the economy, what are they trying to sequester? Money. Kind of. The budget, I guess. Right? They're trying to kind of like gather money and set it aside by taking it away from things. Yes. Oh, you were just.
Sorry.
You're just like people in the back row are all like jazzercise. To isolate or set aside. Okay, so let's talk. So isolate. Isolate is good. Okay, so receptors can be downregulated through sequestration in multivesicular bodies. So let's look at what's happening in this figure. You have your receptor being bound by a ligand, and then you get endocytosis, and then what you actually have happening is further kind of like endocytosing into the endosome, and that you end up with these multivesicular bodies with these vesicles inside the endosome, basically. So the whole entire thing. So you have the further, so you have it here, and then it buds in to the endosome. And so the receptor is sequestered inside the endosome and kept away from the rest of the cell. Okay? And so why would you want to do this? Why would you want to do this? Let's see who's not here. Dennis Clemens. Dennis, where are you? It's like Bueller. Who loves that movie? I'm getting too old. Yes. Yay. Okay, Dennis is not here. What you got from Dennis? Matt. I saw you. Matt. Yeah. But at least you're here.
I was trying to write down.
Okay. So what's the purpose of sequestration in a multivesicular body? So I'm talking about receptors and how they'll endocytose and they'll end up in a multivesicular body. Why? What's the purpose of doing that? What's the difference between doing that and just going to an endosome? Feel free to talk to your neighbors.
So you said that sequestering is isolated
Yeah. From the rest of the cell. So when you're sequestering, you're isolating your receptor from the rest of the cell. Huh? Well, it's basically to compare it to going to the endosome. The traditional thing we've talked about is where you've got endocytosis and it goes in the endosome, right? But here, and then to the lysosome. But here, it's budding in again, and it's ending up in this multivesicular body. So why might you want to do that? Okay, who else? I saw someone raising. Yep.
Isn't it like the cargo?
Does it dump off cargo? So that's but that's similar to what happens in an endosome, right? Where like you've got the acidification and the cargo separates from the receptor, right? So why a multivesicular body? Yes. No? Yes.
Yes. Is it so that, like, because those were in the membrane, is to make sure that they're not going into the membrane and the lysosome where they're actually being degraded?
Okay, so you're getting close. So is it to make sure that they're going to actually get degraded? They're not going to be in the membrane of the lysosome. You're getting close. Yeah. To degrade the receptor as well. Okay, so we're getting in the right direction. These receptors need to be degraded, but why are they not just in an endosome? Yes.
Is it to, like, to prevent signaling while it's in?
Yes. Yes, $1 million question. or $1 million answer, to prevent signaling. So, right, don't forget what receptors do. They have a cytoplasmic tail, and this is why I'm telling you to remember this as we move into signaling. They have a cytoplasmic tail, and the purpose of that cytoplasmic tail is it's going to bind to things upon liganding on the cell surface, and it's going to activate kinases, which will phosphorylate things, and other proteins will come and bind, and they'll activate other proteins, all in the name of activating gene expression, right? That's what signaling is. It's sending a message from the outside of the cell to the inside of the cell. Yes?
So, these receptors were giving sort of other kinds of things, they would be signaling, I'm just trying to understand better.
Yep.
In the
So-
cytoplasm, not inside the endosome itself.
Right. So I know exactly where you're going. So your question is, so if these receptors were not here, but they were up here, right, in the endosome,
Yes.
would they be signaling? Where's their cytoplasmic tail? Is it in the cytoplasm? Right. And where are all the signaling proteins? In the cytoplasm. So what's the difference between this and this? Nothing. So when you're trying to downregulate a receptor to shut off a signal transduction pathway, you want to sequester it so that it can't keep signaling. That the whole purpose is to shut off that signaling. Is everyone following that? Isn't that cool? And actually, HIV, I'm full of all of this information about HIV. My apologies. HIV uses the same machinery for making a multivesicular body. It actually recruits it to the plasma membrane, and that's how it buds out of the cell. Isn't that cool? Because it's budding machinery. Yeah.
I feel like we've heard this in the last class, but what's the difference between the multivesicular body and the endosome?
So the question is, what's the difference between the multivesicular body and the endosome? An endosome, all it is when you endocytose something and then it ends up in this vesicle, right? But notice your extracellular side is in the lumen of the vesicle, and your cytoplasmic tail is in the cytosolic side, right? But in multivesicular body, you bud in one more time, so the whole entire thing is inside this body. It's a vesicle and a vesicle. But your topology remains the same, so things flip around, but the whole thing is inside. Nothing's exposed to the cytosol anymore. So if you're trying to shut off signaling, there you go, right? All those proteins that are involved with interacting with the cytoplasmic tail that send the signal to the nucleus, they're all hidden, they're sequestered, like our budget.
These multivesicular bodies, like these are the same things that come off of the Golgi and come together?
So are these the same thing that come off the Golgi and come together? No.
What was the name of that?
Are you thinking about the things that came off the ER and these vesicular tubular clusters that moved from the ER to the Golgi? Different. That's really just all these things. They're not butting into each other. They're just fusing together. These are these vesicles coming off the ER, fusing together and moving towards the Golgi. Here they're actually butting in one more time. Okay. Any other questions? I saw a hand up. Everyone feeling good? Was that fun? Do you guys like when I call randomly on you? No, I'm sorry. Won't do it anywhere. Okay. So I want you to think about why, not just that this happens, but just why. So why not send the receptor complex directly to the lysosome and an endosome? Because you want to hide that cytoplasmic tail. Very good. Okay, that's that. So then what else do we want to talk about here? So endosomes can store membrane proteins for use when needed. And so this is a specific example with glucose transporters. So normally you'll have, just in a normal cell, you have your insulin receptor, and there's a glucose transporter, and then there's this intracellular pool of glucose transporter that's in this recycling endosome, just hanging out, waiting. And when you have insulin signaling, it'll signal a whole bunch of things, but one of those things is to send a transporter up to the cell surface because insulin signals to bring in glucose into the cell, right? So that's one thing that endosomes can do. Yes?
Endosomes move like in the cell, they'll
So the question is, so if you've got these endosomes floating in the cell, just waiting around, what keeps them from fusing with the lysosome? So what regulates, so that's a really great question that you're going to answer yourself in a minute. What regulates this vesicular transport? What determines whether you're going to have fusion between two vesicles or two membranes? And anyone can answer this. No, we're talking about vesicle fusion. Rabs, right? And snares. And so if you don't have the right pairing of things, the right rab and rab effector, you don't have the right snares, then you're not going to get fusion. Yeah, so that's regulated, and that's a good way to be thinking. Yeah. Okay. Right, so signaling through the insulin receptor stimulates localization of glucose transported to the plasma membrane. So this is regulated. You don't want tons of glucose transporters in your plasma membrane when you don't need them, right? What kinds of cells might have large intracellular pools of glucose transporters? Just, there's all these different cell types, and they all do different things and are in different environments. Which cell types do you think might have lots and lots of large intracellular pools of glucose transporters? Muscle cells, that's one. What else? Give me one more cell type that loves to take up glucose. What cell type is responsible for beer bellies? Adipocytes, fat cells? They like to take up glucose? I don't know if that is really responsible for beer bellies. I think the beer is responsible for beer bellies. Were you going to ask something? No. I thought I saw a hand up. Okay. Good. Okay, so let's talk about secretion, also sometimes called exocytosis. Remember, endo, inwards, exo, outwards. Secretion, not excretion. People always want to say excretion. What's the difference between secretion and excretion? There's some difference. I know this is secretion. Excretion, I think, is removal of waste, right? And this secretion is not usually waste. It's usually this very purposeful secretion of a molecule for a reason. So I'm just correcting that because I hear it over and over again. Okay, so this is, you have ER, Golgi, secretory vesicle, cell exterior. And sometimes you'll go, we'll talk about why it goes Golgi, cell exterior, or secretory vesicles, cell exterior. And that has to do with whether it's regulated or constitutive. Right, so there's two kinds of secretion, regulated and constitutive. What does that mean? What is regulated secretion? Can someone give me an example? Yes.
Isn't it response to the stimulus or the signal?
Right, so it's in response to some kind of stimulus or signal. What's an example? Yes. Insulin. What's another example? Who has hay fever? Histamine, right? I heard someone say histamine back there. Right. And so what's constitutive secretion? What's that mean? Yeah. Well, constitutive gene expression means genes that are already expressed. Take that concept. What does it mean for secretion? Always being secreted. So it's not regulated, it's just always happening. So what kind of molecules might follow the constitutive secretory pathway? What do you think might always need to be secreted? Yes? ATP production. Is ATP secreted out of your cell? I don't know. Maybe. What else? So don't forget so when you have a secretory vesicle or a vesicle headed for the plasma membrane, what's that vesicle made out of? What's it made out of? Huh? Lipids. right? And those go to the membrane and they become a part of the membrane, right? So don't forget, there's this constant exchange of membrane between your plasma membrane and all of these vesicles in ER and Golgi. And so there are certain times during cell cycle when you need to grow your membrane, right? When do you think you might need to grow your membrane? During cell cycle, just think cell cycle. I know what you're thinking. Would you ever need to expand your cell membrane? Yeah. When? When the cells divide, right? You're going from one cell to two, right? So something that might follow the constitutive pathway is lipid, right? Don't forget that. Okay. So the constitutive secretory pathway is the default for all proteins leaving the Golgi unless they're diverted elsewhere. I've said this like 20 times, so you guys all know this, right? Remember, you have ER, Golgi, and then pretty much out of the cell. If you're a membrane protein, you stay in the membrane. This is the black one, all the way through, and you are a membrane protein in the plasma membrane. If you're in the lumen of the ER, you're in the lumen of the Golgi, in the lumen of the vesicle, and you're secreted out of the cell, right? If you have a mannose-6-phosphate added to you, where do you go? Lysosome. So that's a diversion. You know, if you have a transmembrane domain, you're in the plasma membrane. So what's missing from this figure? Where can the proteins go that it's in here, but they don't really highlight it? There should be a number 4. Yep. Right, chaperones, so ER resident proteins going back to the ER because they have the K-Dal sequence. Right. I always love these kind of, I think when you guys are studying, really pay attention to these kind of big picture-y figures in the textbook. This is why I love the textbook, because they kind of have it all in one spot. Okay, so secretory vesicles contain specialized products that are secreted in response to a signal. This is the insulin and histamine and all that. And you have them coming out of the trans-Golgi network, and then they're fusing with this secretory vesicle. So you can think of it as multiple small vesicles fusing together to create this secretory vesicle. And then what happens is you have some vesicles bud off and go back to the Golgi to concentrate it. So they're all kind of coming together in this very large vesicle, and then vesicles containing nothing without cargo bud off. So it's almost like membrane is budding off and heading back to the Golgi, and then you end up with this mature secretory vesicle that's full of a lot of whatever needs to be secreted. So, do you have a question? Is that an example of pinocytosis? Well, pinocytosis is coming in from out of the cell, right? But it is in a way kind of like budding off of your secretory vesicle with nothing in it. But pinocytosis has some things in it. So it's not well understood, and it says it right in the textbook, but can you guess or hypothesize how cargo is correctly packaged and concentrated in secretory vesicles? We've learned a lot about packaging and targeting of different kinds of cargo to different places. So I want you guys to take a minute to chat with your neighbor and come up with a mechanism of how proteins that are supposed to be packaged into secretory vesicles are packaged in. Because remember, you have your secretory vesicle, that's your regulated secretion, and then you've got your constitutive, which is just straight up no regulation. So there has to be some differentiation between what goes just out of the cell and what hangs out in the secretory vesicle until it's needed. And so what kind of mechanism, think about how you learned about how different proteins go to the lysosome, different proteins go to the ER, different proteins make it to the ER from either direction, and what kind of mechanism can you suggest for packaging of secretory proteins into secretory vesicle. So take a minute, discuss, and I'll circulate and clarify. What? Well,
Does classrooms like fall off after it gets pretty low level?
so clapper, yeah, so after you form your vesicle, there's uncoating.
Okay.
Yeah.
I thought it was just kind of until
Okay, so are you guys ready to discuss? Okay, so who has a hypothesis they want to share? Yes.
Is there like maybe some sort of like tag or signal that keeps it in the vesicle stated as needed, or one that just says, okay, this can go out continuously?
So something about maybe there's a tag or a signal that keeps it in the vesicle. Well, so you're on the right track, but the difference is it's about packaging into the vesicle.
Send it to them.
Right, okay, right. So some kind of signal or tag that sends it to the vesicle. What else? Yes.
There maybe is some kind of like recognition of the specific signal that needs to be packaged, so they're all like brought to
Right, so some kind of recognition of the signal that needs to be packaged. So what have you guys learned already about the recognition of different signals to be packaged? Yes. So a receptor, right? So it's exactly what we've been talking about, like the KDL receptor, mannose phosphate receptor. In all these situations, the protein has some kind of sequence or something, a tag or something that's recognized and either modified or bound to by a receptor or something, but in the end, it's bound by a receptor that specifically packages it into a vesicle. right? And so why would it be any different? I mean, it's not, no, according to the book, I don't know, the book is a few years old, so maybe since then it's known, but it sounds like it's not really known. But there's probably some kind of sequence or something that's specific to getting into the secretory vesicle. Why would the cell reinvent the wheel? Why would it reinvent the wheel? Right? Any thoughts? Anyone get anything else? That's good. It's good that I'm hearing what I want to hear. I'm training you well. Okay. So this is just showing release of insulin from a secretory vesicle in a pancreatic cell. Well, this is the picture, but this is what it would look like. Okay, so this is all the insulin. And here's the vesicle. That's the membrane of the vesicle, and it fuses, right? So here it's not actually fused yet. It's just waiting. But here it's already fused. So this membrane becomes this membrane, and it just releases the insulin out of the cell. That's what your cells are doing right now. Okay, so in another thing that happens, you've got your secreted products that are concentrated and processed in secretory vesicles. We talked a little bit about processing, and this means usually some hormones and cytokines and different kinds of proteins that are going to go out and do something. They're usually in an inactive form, a pro-protein, right? And then usually something needs to be cleaved off for it to be active. And so usually it stays in this pro form until the secretory, sometimes in the Golgi and then often in the secretory vesicle, it'll be processed. So when it exits the cell, it'll be in its active form. Why would you want to do this? Why would you just cleave it in the ER? Why are you waiting for the secretory vesicle to cleave it? Not you. Why does the cell do this? Yes?
It might be like harmful if you cleave it earlier, because maybe like the acid hydrol. Yeah, that one. Because it like has to be in the pH.
So you're comparing it to the mechanism of regulation of the activity of acid hydrolases, right? Great. So remember the acid hydrolases, and that's a good thing to compare it to, because the acid hydrolases are harmful to the cell and shouldn't be active until they get to where they need to go, which is the lysosome, and so they're regulated by pH. And a lot of these pro-hormones or pro-proteins, you don't want them active either because they're harmful, or maybe they're going to signal, right? So a lot of hormones, they signal certain things, like the binding hormone receptor and activate gene expression. You don't want that to happen in the cell that is being made in, right? You want to get out of the cell and get into the next cell and do what it needs to do, right? So that's the right way to think about it. Any thoughts about that? No thinking. Okay. So you might be thinking, but how are they just a plasma membrane but not fusing? And so we were talking a little bit about grabs and snares. And so this is what happens with the snares. The secretory vesicles are at the plasma membrane, and they have their snares like partially wound, but not completely. So you can think of it as like the gun. I mean, I should talk about guns, but like, you know, the gun is like cocked and loaded and ready to go. And as soon as the signal comes, then the snare is fully wind, and then you have fusion and release of the contents outside of the cell. So they're waiting for that signal, but they're ready to go. And that's kind of like what we saw here. They're kind of just hanging out right here, nearly fused, but not quite. Okay? So this isn't that, I always love this picture because like it just screams, this is how I feel during hay fever season. So this is a mast cell. Your mast cells are the immune cells that respond poorly or very well, depending on your outlook. Unstimulated mast cells contain all these vesicles. They're packed full of vesicles containing histamine, right? Histamine, not your friend, right? And then, so this is the stimulated mast cell, and you can see, like, they're releasing, the vesicles, well, releasing the contents all over the place. That person probably didn't feel very well. Luckily, the cells were taken out of the person prior to this experiment. And so endocytosis, I want you to think about endocytosis and exocytosis as this thing that's constantly, both of them are happening at the same time, and that it affects the membrane size and composition for the cell. And so sometimes you're going to want to grow your cell, and you might have some more exocytosis to bring more membrane up to the plasma membrane, but generally it's that constant back and forth that's maintaining membrane size, right? If all you had was tons and tons of secretion, then you'd have like this gigantic cell, right? Something came in to kind of keep to equalize the membrane. Okay. And like what I said before, why would you want to increase your membrane size? Well, in cell cycle, you do have to go from one cell to two, right? So that's useful. And then also, with phagocytosis, you also have these huge pseudopods kind of coming up over the bacteria that's being phagocytized, and that's a huge amount of membrane that's coming up and over. And then I believe also with repair of plasma membrane in cells that have a lot of stress, such as muscle cells. Okay, so we'll talk briefly about polarized cells. These are cells which have top and a bottom, different sides. They actually have different sides with different functions, right? Like epithelial cells, for example. And so they can secrete things at different sides because different sides are exposed to different environments. So they might secrete something different at the apical side versus the basolateral side. So that can happen in two different ways. Sometimes there'll be this direct sorting of proteins. They'll go to where they need to go. They'll have some sort of cytoplasmic signal in their cytoplasmic tail, and that'll send them to the right membrane. And that's what's showing over here. And then sometimes there's more of this indirect sorting going on, where you'll have it going to one side, one membrane, and then you'll have it endocytosed. So that there'll be endocytosis to endosomes, and then be recycled to the correct membrane. Okay, that's it. Not really. That's it for the secretory pathway. And so for the next 2 days, we will be talking about more. Don't forget, the cell is not just made up of a nucleus, ER, and Golgi. Well, ER and Golgi and lysosome, but there are other organelles, and those other organelles are going to have proteins that need to work in them, just like acid hydrolases work in the lysosome, and BIP works in the ER. So different proteins need to go to different places, right? So these are the different kind of categories of transport for proteins. You can have vesicular transport, which is what we just spent the last few weeks talking about. Then there's gated transport, and that is what we're about to talk about, where we're moving things from the cytosol to the nucleus. There's no vesicles here, bless you. There's that just direct binding of proteins by transporter proteins that help them to get through a pore, in this case the nuclear pore. Then there's transmembrane transport, and this uses these membrane translocators. Remember the translocon in the ER? And then the other example would be the mitochondria, which we'll be talking about. I won't get to that till Wednesday. So what determines the correct address for any given protein? Some kind of, I heard someone say tag, signal sequence, right? So there's some sort of identifier in the protein sequence that's going to determine where the protein needs to go. So that's a concept that kind of holds for everything. Okay, so here we're going to start talking about transport between the nucleus and the cytosol. So why would proteins need to enter and exit the nucleus? What kind of processes in the cell might require proteins to go in and out of the nucleus? I'm going to try my random mark. DNA and replication. So proteins that participate in DNA replication, right? Because where are they translated? In the cytosol, and then they need to go into the nucleus. Okay, there's one. What else? Yes.
Things that affect gene expression.
Right, proteins that affect gene expression, such as transcription factors. That's a big one, and as we start talking about signaling, we will talk a lot about that, how all these signaling through receptors is really just doing something to send a transcription factor into the nucleus to activate gene expression. Yep. And so what kind of proteins would you expect to be coming out of the nucleus? Yes? RNA transport, right. So RNA, right, it needs to get out of the nucleus in order to be translated. And there are all these different proteins that bind to mature mRNAs that help to get it out of the nucleus. What else do you think would be exiting the nucleus? What else? What else? I'm sure there's other things.
Maybe like just anything that needs to be degraded outside the nucleus.
Something that might need to be degraded outside of the nucleus. Well, you're making the assumption that nothing could be degraded inside the nucleus. I don't know. I actually don't know. But maybe there might be a reason to export something out of the nucleus to be degraded. I'm not sure. Yep. Like they need to come back out. Transcription factors, do they need to come back out? I think sometimes they do come back out. Would they need to be degraded? Well, transcription factors do have a certain half-life, and that's related to their activity, because obviously when you want to turn off gene expression, you would want them to be degraded. Good example. Usually have to have a little context to care about things, I guess. Okay, good. So macromolecules enter and exit the nucleus through nuclear pore complexes. So there are these pores in the nucleus. You can see them here, right? So there's your nucleus, and remember it's continuous with the ER, and there are these pores that things enter and exit through. And the number of nuclear pores change depending on the conditions. So it's not just like there are 10 nuclear pores. It depends on what part of the cell cycle things are, or how much gene expression, or whatever. So when do you think you might need to have a high number of nuclear pores? Lindsay? Lindsay? No, Lindsay? I feel like I'm being left at the altar here. Okay. No, I'm just kidding. Right, if you need a bunch of stuff to get out or get in. So what's an example of when you would need a lot of things to come out of the nucleus? Right, so if you're signaling that's activating gene expression and lots of RNA needs to come out, so that's a specific signal that's going to result in lots of gene expression, right? So you're going to have, in the unfolded protein response, you'll get expression of lots of genes that are involved with the unfolded protein response, such as chaperones, right? So in any sort of response to something where there's a high level of gene expression induced, you'll probably also be inducing more nuclear nuclear export related things such as maybe nuclear pore complexes or export and stuff like that. Good. Okay, so active transport is required for larger proteins to enter and exit the nucleus. Yes.
I'm sorry, were you saying that the nuclear pores can be like they make more amount of strength?
They change. Yeah, their nuclear pore numbers fluctuate based on need. That's surprising, shocking,
I
amazing.
just thought there was like
It is what it
number
Static.
was always there.
Yep, I was surprised to read that too when I was first reading that. So you need this active transport for large things like proteins. So small molecules that are 9 kilodaltons or 9 nanometers or less in size can just diffuse right through it. But larger things such as DNA, RNA, polymerase subunits, those are 100 to 200 kilodaltons. Way too large to just diffuse through. So you have this active transport. And then ribosomes are pretty large too. So why else is active transport more desirable than diffusion? Yes?
You wouldn't want just any sort of small molecule to go in your nucleus. Probably not a good thing.
You wouldn't want any small molecule just heading into the nucleus. It's not a good thing. Why? You're right. Why? Well, yep.
The center of the cells, and then nothing is wrong with protein.
We don't want our proteins jacked up. Right. Right out of the textbook. Albert said that. No, but so give me an example. Like so what I'm trying to get you guys to tell me is that yes. Right. Okay, so something about binding to what? DNA, right? So what is gene expression? It's transcription factors binding to DNA and inducing transcription of certain genes. And so sometimes that's completely regulated by whether or not something goes in and out of the nucleus, is able to get into the nucleus, right? The other thing is you don't want any old RNA coming out of the nucleus, right? You don't want RNAs that aren't properly spliced or aren't capped or whatever, just randomly coming out of the nucleus. You only have mature RNAs that are ready to function. Good. Yes.
So technically, this is the active transport the same when we have like viruses like HIV and stuff like that, because that's something we didn't get out, with all the exons and still it's
So the question was, is this all disabled when we have certain viruses like HIV that need to get their genomes in and their genomes out and have all these introns in there? And so the answer is viruses have evolved their own mechanisms to get around all this, and they usually involve having proteins that will bind to important or exporters and help to get the genomes out of the cell. Viruses are really okay. Good. Okay, so you might be saying to me, show me the nuclear localization signal. I need to know how are these proteins getting into the nucleus. Here it is. Proteins that contain a nuclear localization signal localized to the nucleus. And so basically it's a lysine arginine rich region in a protein. So anytime you think that a protein might be nuclear, you can look at the sequence and say, oh look, there's a nuclear localization signal, or there's not. You know, it's good. So this is showing, short sequences enriched with lysine and arginine tend to be nuclear localization signals. Don't memorize this, but understand lysines and arginines, nuclear proteins, okay? So this is just an experiment where they were looking at the localization of T antigen, which is an SV40 protein, and it has a nuclear import signal, or an NLS. And so this is the nucleus. So it's just showing it's located in the nucleus. And what they did is they mutated this nuclear import signal. All they did was change one amino acid. And now, is it nuclear? No, that's what cytoplasmic. So these circles are the nucleus. See how the white is all around it? And so it's suddenly cytoplasmic. And that one mutation prevented the T antigen from getting into the nucleus because it can no longer recognize the correct machinery. Okay. So I have a question for you. So if you were studying nuclear import, so if I gave you an exam question and I said, you're studying nuclear import and you see this sequence and I give you a sequence that has lots of lysines and arginines, what would you do to prove that your protein truly was imported into the nucleus and that it was dependent upon the lysines and arginines that you're seeing? Yep.
Tag thing, where it's like trying to trace the picture, and then if you had just like unchanged chain or just a whole bunch of like lysines like that, and switch them, you'd be able to tell stuff like
Okay, so you can actually fluorescently label different proteins and either have the wild type nuclear localization signal or what? Huh? Or you can change it up and see if it still goes there. Yep. Stacey, you raising your hand?
Well, I was just gonna say you could mutate another protein to make it work.
You can mutate another protein to see. Okay, so that's another question. So what if you wanted to purposely send the protein to the nucleus? You can add this lysine arginine rich region and see if that'll take it to the nucleus. Or what if you were studying your protein and you knew it was nuclear and you were but it didn't have this stereotypical lysine arginine rich, but you thought maybe it had this non-canonical nuclear localization. Non-canonical means not the typical one, slightly different. What can you do? You can do a similar kind of thing, right? You can take that sequence and put it on something and see if it goes into the nucleus. So these are the way people do research. Yeah.
So you're saying the sequence is lysine or arginine or whatever it was. What if you change the lysine to arginine? Would that change it drastically? Or
So the question is, if it's a lysine arginine rich sequence and you mutate the lysine to an arginine, how might it affect nuclear localization? What do you guys think? Do you think it would be nuclear? If you think it would be nuclear, raise your hand. If you think it would be cytoplasmic, right? You think it would go cytoplasmic? If you change the lysine to an arginine?
Yeah, because it's not.
Well, so this is the thing that, so,
Does it have to be?
right, so you're thinking it has to be exactly this. And it has to just be lysine arginine rich. So there's no specific, it's not like lysine,
Or
lysine,
does
lysine,
it
arginine,
have to
arginine.
have like this many lysines and this many arginines? Because where if you're adding too many, then it won't.
So the question is, does it have to have a certain number of lysines and arginine? It's just lysine arginine rich, and I think as long as you have like, I don't know, there's probably a certain number that's ideal.
Yeah, because where if you have too many.
But his question is, what if you change a lysine to an arginine? So whenever you're making mutations, this brings up a good point. Whenever you're making mutations, you want to think about what you're doing. Because if you were studying this, and you said, hey, I'm going to mutate this lysine to an arginine, it might not tell you very much, but if you turned it into an alanine or something like that, something very, very different, then you might get more information out of it. Any questions? Okay. Okay, so here, this is just from an experiment from the book where they used gold spheres, literally gold spheres, coated with peptides containing nuclear localization signals. So they coated this gold with lysine arginine rich sequences. And so this is the nuclear envelope. So this is just some EM, electron microscopy, and they took pictures. And so you've got your, this is your nuclear envelope. And so in 10 minutes, your spheres are up here. 30 minutes, they're down, kind of entering the nuclear core. And then 40 minutes, they're kind of on their way, they're more in the core. 50 minutes, you're starting to see some of them on the opposite side. So this would be cytoplasmic, this is nuclear, right? And so you can kind of watch them passing through the nuclear core. Is that cool? Yeah, kind of amazing you can do that. Okay, I think we might stop here because I'm hungry. And we'll continue on Wednesday. And on Wednesday, we'll finish this and we'll finish the mitochondria.